Today I would like to talk about molecular epidemiology of ESBL-mediated AMR in companion animals, E. coli isolates, with the specific focus on Vienna and on companion animals such as dogs. This is the study we just published this year in Journal of Antimicrobial Chemotherapy. And I would like to start actually first with a quick reminder why we need to monitor ESBLs in general. So I'm pretty sure this audience already knows that beta-lactams are among the most used antibiotics. They were discovered. It's the first antibiotic discovered by Alexander Fleming in 1928, and already in his acceptance speech for the Nobel Prize a few years later, he acknowledged that there is a problem with resistance, and in large part it's driven by the overuse. Unfortunately, or fortunately, beta-lactams are antibiotics that are the most used. So you can see this very well from the this sketch by the WHO AWARE classification of antibiotics, which is split in access, watch, and reserve groups. And you will find beta-lactam antibiotics in each of them. So access are those that you normally would get when you go with uncomplicated infections to your primary care provider. Watch antibiotics are those that are reserved for specific indications, specific syndromes for which others cannot be used, and they have a higher risk of antimicrobial resistance development. And reserve antibiotics are those that usually are reserved to clinical context and are used in case of antibiotic-resistant infections, resistant to other infections. So you can very well see that beta-lactams are very well distributed across this entire list, and beta-lactams of the second and third and fourth generations, they're all in the access and watch groups. And we specifically focus on beta-lactams with extended spectrum beta-lactamases because they are threatening these groups of antibiotics. And these groups of antibiotics are very important because they are used for the treatment of pretty much most of infections due to their very good profile in terms of pharmacodynamic, pharmacokinetic, tissue distribution, and so on. So they're very important. And a little reminder about ESBL. So there are several groups of beta-lactam antibiotics. Penicillins you probably are familiar the most with, for example, penicillin G or ampicillin or amoxicillin. Those are very often that you get from primary care providers. It also includes monobactams, cephamycins, and cephalosporins that can be further divided into 4 generations. And you probably also are familiar with quite a few of them, like third generation cephalosporins, such as cefotaxime. These beta-lactam antibiotics can be inhibited by extended spectrum beta-lactamases, specifically penicillins, monobactams, and cephalosporins up to 4th generation, like usually 3rd, but some people include the 4th generation, but not cephamycins and carbapenems. Carbapenems are very important. I will mention them again, also because they are used for infections caused by ESBL Enterobacteriaceae. So this extended spectrum beta-lactamases, most common of them are those encoded by TEM, CTXM, and SHV beta-lactamase genes. And in turn, these beta-lactamases can be inhibited by beta-lactamase inhibitors, such as clavulanic acid. So this is basically the definition of ESBLs. Those are organisms that produce beta-lactamases that are able to inhibit penicillins, monobactams, and cephalosporins, but in turn are themselves inhibited by beta-lactamase inhibitors. So now that we know what ESBLs are, we can move on to Enterobacteriaceae and why ESBL-producing Enterobacteriaceae are an important topic to study. Well, first of all, they're very common colonizers of gastrointestinal tract, and some of them cause hospital and community-associated infections, such as urinary tract infections, respiratory tract infections, bloodstream infections, wound infections. Very often they are carrying mobile genetic elements such as plasmids that may have other resistance genes and may be co-resistant to other important groups of antibiotics such as carbapenems and fluoroquinolones. And we see an increasing rate worldwide, but also in Europe. You can see on this slide from the map from the European disease prevention, disease and control authority, CDC, the prevalence. And you can see that in some regions of Europe, especially in the south and in the east, the prevalence of ESBL Enterobacteriaceae can be up to 25%, in some cases even up to 50%. A word of caution here when we talk about this incidences, this is something that is reported to CDS, and those are monitoring programs that are stipulated by the European Commission. So currently the monitoring is focused on invasive and non-invasive bloodstream infections in hospital environments. So hospital isolated bacteria, and in veterinary medicine are those from food-producing animals, which basically leaves us a large part of One Health spectrum outside. So this was part of the motivation why we wanted to focus on dogs, because One Health approach is very important to antimicrobial resistance, but at the same time, it's very much skewed to hospital isolated bacteria from the human health and food-producing animals in the animal health, whereas other areas of One Health are not as strictly monitored. So pets, we know they can carry bacteria, antibiotic-resistant bacteria, so they can act as carriers and sources of infection. And dogs occupy a very unique niche in the One Health spectrum because they're in a constant and close contact with their human owners, with other animals, with the environment. So we thought that it's an interesting and understudied subject to look at antimicrobial resistance. And it may not be such a negligible topic if you consider that in Austria alone, there's almost 1 million of dogs, and of them 55,000 living in Vienna. And the last study that we had in Austria about ESBLs and Enterobacteriaceae in dogs was in 2012, and the prevalence was pretty low, but it was also in a very remote region in Austria, so 2.2%. Basically, we didn't have any fresh data. So that was what motivated us to do this study. Essentially, we had a very simple question. We wanted to know what is the prevalence of ESBL-producing Enterobacteriaceae among dogs. And these dogs are those that come to our university clinic for different reasons. Some of them are healthy, some of them come for vaccination, some come for treatment. But overall, it gives a good representation of dog population here in Austria. So a few words about our methodological approach and studied population. We had 88 dogs coming to our clinic. Our clinic is located here in the northeast of Vienna, on the border of 2 districts. And you can see in each of the districts the number of animals that we had from each of them. So it was fairly well distributed, but of course skewed to the areas that are most physically close to our clinic. So the 2 districts in the north, in the east, and the surrounding federal state of Lower Austria. We had a convenient sampling strategy, which means this is random, but not really representative of the whole Austrian population. We collected fecal material. Our animals were between 1 and 13 years old. A word of caution, our clinic specializes in inflammatory gastrointestinal diseases, so almost half of the animals who visited the clinic had an inflammatory gastrointestinal disease. So that was our bias in our convenience sampling strategy. And about 1/3 of them received beta-lactam antibiotics 6 months prior. We did the typical methodological approach, selected the bacteria were plated on selective agar, McConkey agar was supplemented with cefotaxime. We did a combination disc diffusion to check for ESBLs. We confirmed them by VTEC. We used VTEC and MALDI for species identification, then whole genome sequencing, and phylogenetic analysis, population structure, antimicrobial resistance genes, virulence genes, and so on. And finally, we did some phenotypic assays with the selected isolates. So to the first result, sizable proportion of dogs attending our clinic were actually ESBL positive. You can see here on the map in red are the number of dogs that were positive out of the total number of dogs from those areas. Overall, we found almost 15% prevalence of ESBL-producing Enterobacteriaceae in dogs attending our clinic. Most of the genes that encoded these ESBLs belong to CTX-M1, CTX-M15, and TEM1B, which is also very much in line what we saw from the literature published on this topic in Europe. And while this prevalence was significantly higher than European average of 6.2%, and especially much higher than the last data we have for Austria and Tyrol, this is more or less comparable to another large metropolitan area such as Paris. I mentioned that some of the animals have gastrointestinal disease, others also received beta-lactam, so we did mathematical modeling to see if there was any association with any of those factors and ESBL status. Our sample size is pretty low, 88 animals, so we did not find any statistical association with age and inflammatory gastrointestinal disease, but the p-value for the beta-lactam antibiotics use was kind of borderline, so it's not significant, but there may be an indication of a big correlation with the beta-lactam use, and this is something we saw in our other studies and studies of others when antibiotic use is associated with ESBL status. Then we focused on this specific ESBL producing isolates and characterized them further, and about 1/3 of them were phenotypically multidrug resistant, and if you look actually at their genomes, over half of them were multidrug resistant, so we have fairly high rates of multidrug resistance in these ESBL isolates. Another important phenotypic resistances were ciprofloxacin, so fluoroquinolone resistance in almost half of them, trimethoprim, sulfamethoxazole, and tetracycline. But luckily, we did not find any carbapenem resistance, which is a positive finding, considering that carbapenems are beta-lactams that are used for treatment of ESBL infections, and something that is being very closely monitored at the moment. So it's a good sign that as sentinels, in a way, our dogs do not appear to have carbapenem resistance. We found a high number of plasmids in our isolates that may, of course, facilitate the spread of antimicrobial resistance genes. And then we looked at the population structure. So for this, we built a core genome neighbor joining phylogenetic tree using our isolates, and pretty much all isolates we could obtain from GenBank from dogs. In total 129 canine isolates. You can see our isolates here highlighted in yellow. We aimed to have as much representation in terms of the country, of the host, of the isolation source. And you can see that it primarily splits into two large clades. Clade 1 primarily contains European samples, and clade 2 contains North American samples. And if we look closer at Clermont typing within them, we find actually quite a lot of isolates that are associated with human-associated isolates. So we found, for example, in the North American clade, two isolates belonging to B2 phylogroup, which is associated with extraintestinal pathogenic E. coli in humans. And in the European clade, we found several isolates from phylogroups C and A, which are also common intestinal pathogenic E. coli in humans, as well as D. But also we found quite a few of them that are more associated with animals and the environment. So we looked a little bit deeper into the sequence types. We performed multi-locus sequence typing. And interestingly, we found a very diverse population structure. They were pretty much different. Each isolate had a different sequence type, except these two, which belong to the same sequence type, which is associated with animals, but they were actually different isolates. They were not identical, which probably suggests some kind of circulation at the regional level of this animal-associated sequence type. Both of them actually were quite encoded quite a high number of enterotoxins. Then what also drew our attention was the fact that a third of the isolates also contained isolates also belong to sequence types from human-associated sequence types. So ST131 and ST38 are very famous high-risk extraintestinal pathogenic E. coli pandemic clonal lineages that combine antimicrobial resistance and virulence. And ST141 is an emergent expect clone, which is associated with community-associated urinary tract infections. So we looked closer at each of these isolates, and again used the same approach. We built phylogenetic trees. In this case, we pulled the E. coli specifically belonging to these sequence types, those we could get that would qualify certain quality criteria, but we aimed at as wide representation as possible, so from different countries, different host species, different diseases. And we were able, for ST141, find 42 whole genome sequences and build this phylogenetic tree. So our isolate here highlighted in yellow comes from a 5-year-old mixed breed dog with irritable bowel syndrome disease, and this isolate was multidrug resistant to beta-lactams, sulfamethoxazole, and aminoglycoside, and belong to this extra-intestinal pathogenic E. coli phylogroup B2, which is common in humans. and associated with community-associated urinary tract infections. So this isolate was genetically very much different, as you can see by the length of the branch, from all other isolates, but nonetheless it clustered with human isolates, clinical isolates from France and Poland. Then we looked at the isolate that belonged to sequence type 38. This isolate came from a 5-year-old Labrador from Lower Austria that suffered from a severe case of acute hemorrhagic diarrhea syndrome that resulted in bacteremia. And it was resistant to beta-lactams and quinolones, belonged to the D1 phylogroup, which is human-associated. It's a high-risk international pathogenic clone. So if you look at here at clustering, opposite to the previous case, it was very much genetically similar to an isolate from United States, human isolate from urinary tract infection. And if you look a little bit wider, it's actually nested in the in the general cluster of predominantly poultry and bird samples from Northern Europe, from the Netherlands, Norway, Sweden, and Iceland, and one environmental strain from Sweden. So this kind of, in my opinion, highlights this truly pandemic nature of this sequence type, that they really are able to colonize various environments, various hosts, such as humans, dogs, but also birds, and perhaps even hint at birds as transmission vectors, although of course we cannot really prove it with just this phylogenetics. Finally, we looked at ST131 isolate, which is also highlighted here in yellow. We were, because it's a very much studied pandemic lineage, we were able to obtain 133 whole genome sequences. And here I'm just showing you part of this tree, because otherwise it would simply fit on the screen. This isolate came from a 3-year-old Chihuahua with necrotizing hepatitis from Vienna. It's of course resistant to beta-lactams, but also quinolones, another very important group of antibiotics from the AWARE classification of WHO watch. And if you look at the phylogenetic tree, you find that it's nicely nested among isolates from other pets, cats and dogs from France, and one human isolate from Denmark, but it is actually forming its own subcluster. It's quite genetically distinct from all of them, which probably suggests that this pandemic human-associated sequence type has a degree of pet specialization, because it really fits in European pet samples. Then we looked at the virulence-associated genes in all of these isolates. We found 164 of them, belonging to 6 functional categories. Most common were those associated with adhesion, abilities to survive environmental stress, effector delivery system genes, and iron uptake. If you may remember in the beginning when I talked about sequence types, there were 2 isolates that belonged to the same sequence type. So they had this EST1 enterotoxin, and actually both of those animals had enteric disease. We found hemolysin in the isolate that belonged to pandemic sequence type 131. And in the emergent clone 141 isolate, we found the highest number of virulence-associated genes, 102, including in 3 different exotoxins: hemolysin, cytotoxic necrotizing factor 1, and colibactin. Because we see that these two strains encode hemolysins, it was very easy to test if they're really hemolytic, and indeed they were. When we just strict them on blood agar, you can see that ST141 and ST131 strains form very nice hemolytic areas on the blood agar. We also noticed that a large number of these isolates, pretty much all of them, the largest perhaps number of virulence-associated genes that all strains encoded belong to the functional category of adherence. So we decided to look a little bit closer there and stained all of these isolates for cellulose and curly fiber production at ambient temperature and at inner body temperature. And the reason we do this because very often this virulence-associated factors, curly and cellulose, are produced at ambient temperatures because they facilitate the attachment of bacteria to surfaces. But very usually this is not happening at elevated temperatures. So we found that actually 9 out of 13 ESBL strains were able to produce curly at 37 degrees, and 5 out of 13 were able to produce cellulose at the body temperature, probably suggesting that this also may facilitate host colonization and environmental spread. So I'm at the end of my part, and just to sum up, we see that a sizable proportion of dogs in Vienna carry ESBL-producing E. coli, almost 15% of them, and about half of them are genotypically multidrug resistant. Some of these strains combine virulence with resistance, and actually a fairly large number of isolates belong to human-associated pandemic lineages. Our phylogenetic suggests that it seems more likely that dogs act rather as spillover hosts rather than reservoirs. But of course, this spread may be bidirectional, considering the very close contact with humans and the environment. Antibiotic use may select for ESBL carriage, and we believe that the ability to thrive in diverse environments may contribute to colonization success mediated by curly production at ambient and body temperatures, which of course brings all of us to the point that we should not underestimate the transmission risk in the One Health context, and the surveillance and monitoring frameworks should really adopt the One Health approach to antimicrobial resistance monitoring. And with this, I'm really at the end of my presentation.
For inviting. So I'm going to be talking today about the air as an enriched reservoir of hidden AMR diversity. So to start with, let's begin with the definition. We're going to be talking about the aerosols. Basically, that is defined as a tiny solid particles of liquid droplets suspended in the air, which may originate from a wide range of natural sources, including sea spray, desert dust, volcanic eruptions, and sea spray. As well, a substantial proportion of aerosols can come from human activities, such as fossil fuel combustion, industrial process, agriculture, biomass burning, and urban infrastructures. Many of us have experienced those days when the air feels dense and difficult to breathe, right? This is often a result of elevated concentration of aerosols and other airborne pollutants. Here is one dust event that took place in Barcelona a few years ago. So this does not come as a surprise that this dense environment will have an impact on human health. And because of that, there are an extensive monitoring networks of air quality monitoring station that is established across many regions. Here comes only the Europe map, but it's established through all the world. Within this air quality monitoring stations, the concentration of the particulate matter is measured, and this based on the concentration of the particulate matter, the quality of the air is presented. So this is the rather quantitative method measurements, right? But what about the qualitative characterization of those particulate matters? So here is a device that is used within this air quality monitoring networks, so-called high volume sampler, and the filter that comes out of this device only after 24 hours of sampling. Just imagine this is something that we are breathing more or less in a few days. Classically, the elemental and chemical composition is analyzed, providing data on concentration of the PM that we have seen previously in the map. However, the recent research has shown that this data, these same filters, also can trap the environmental DNA from a wide range of organisms, including plants, birds, insects, mammals, and fungi. Therefore, by applying next generation sequencing and metabarcoding techniques, researchers can now identify collected DNA fragments, revealing the biodiversity of the surrounding environments. In turn, in a sense, turning aerosol composition data into the tool for wildlife tracking and ecosystem monitoring. So if we can see those, and this is providing a tools for wildlife tracking and ecosystem monitoring in a global context, a One Health approach. So if this filter can capture DNA from a larger organism, what about tiny ones? Could they also reveal the invisible part of the air? So if we're going to be talking about the life in the air, so we're going to be talking about the biological part of those aerosols, and the definition of those are the bioaerosols that consist of life and dead microorganisms, and the part of those of them. Just imagine we take the only 1/8 part of the same filters that we have seen previously and put it on the agar place. And here we get such enriched growth of different bacterial and fungal strains. And this only counts for, this only counts for 1% of culturable organisms out of all detectable or identifiable ones. So when we compare metagenomic data from this high-volume sampler device implemented in the air quality monitoring station with the one that is references device, so-called SAS equipment, what we see that the quite a good proportion of species are overlapping, but also we detect more species using high-volume sampler, including those that have a potential impact for human health, demonstrating that those bioaerosols may possess a potential threat to environmental and human health. Comparable data that we obtained by those two sampling devices demonstrate how traditional air quality equipment, when combined with the molecular tools like metagenomic shotgun sequencing, can provide valuable insights in the hidden diversity of airborne microorganisms. So we have quite a long experience in working with in bioaerosol research. Our group started working with it already 10 years ago, and we have shown that even at 3,000 meters altitude, the air is enriched with microorganisms. During our flight campaign that took place in Japan, a high-volume sampler was installed inside the aircraft, and air sampling began once the plane reached 1,000 meters, continues up to 3,000 meters high. What we found was that the diversity of the microorganism from the flights are kind of similar to the one, so the diversity from the flights, from the sample from the flights was quite similar to the one that we collected in the ground level. Even more striking was the observation of the of clear seasonal patterns. The blue is marked the February samples, when the white background showing the samples from the April. This pattern actually reflects the origin of the air masses, which above the planetary boundary layer are no longer influenced by the local source, but rather transported from distant regions, sometimes from up to 2,000 kilometers. This is what we have seen in our PNAS study using high split models. So this highlights that the atmosphere can be as a vector of full microbial dispersal, enabling the long-range transport of microbial life. We are using as well the culturomic for our studies, and here to test how airborne bacteria respond to antibiotics, we use the disk diffusion on the organism we cultured. Most of what we isolated came from those three genera, such as Bacillus, Micrococcus, and Lactobacillus. And interestingly enough, these groups barely showed up in our DNA-based analysis from the same filters. And this actually highlights how culture and DNA-based methods really complement each other. Each captures what the other misses. When we look at antibiotic-resistant profile, most of the detected organisms showed the resistant. Of a particular interest is the Micrococcus luteus that showed quite a strong multidrug resistant profile. Unfortunately, we don't yet know the mechanism but how this resistance is happening, but if it's linked to the plasmid-borne genes like those described in the literature, it could potentially spread resistant to other bacteria, but still we don't know well enough the mechanism. To say that we are working with the bioresols, and the bioresol research in quite a challenging nature, because the air is a ultra low biomass samples. To give you a sense of a scale, when we compare DNA concentration with the soil or ocean samples, the air there can be up to 6 log differences microbial biomass. So that means that we need very efficient sampling strategies in order to get sufficient material. On top of that, the biomass in the air is not stable. It can fluctuate in cycles, often during the night, and has events have a very strong impact on the concentration of the bioresols. Another part is that air, per definition, is one of the less studied type of samples. In this graph, we see that approximately 48% of all out of all detected species are unassigned in the air. So saying that we know quite little. But the technology is already there, and if in our first study where I was discussing the diversity of air microorganisms at high altitude, we implemented metabarcoding approaches. Now we are able, and the technology is there, and we are able to implement the shotgun metagenomic sequencing for characterization and identification of air microbiome. Here comes our study. We performed here in Barcelona. During the whole year, we were collecting those samples, and here is a top 40 of organisms we get. This is something that the majority of organisms that we detect there are quite normal, and we detect them normal, and then other groups as well shows that they're present normally in air samples, such as Pseudomonas, different type of Syngmonas, Deinococcus, some fungi, particularly Basidium pollunas. So we even picked some plants, saying that basically confirming the data from others, saying that the filters has a huge potential. We also detect some species that do have impact on human health. Particularly, we do have detect Salmonella, Enterica, Clostridium difficile, Escherichia coli, different type of Streptococcus. Those are normally found in the air. But the main advantage of shotgun metagenomic sequencing is not only the capability to detect the organism, but as well try to see their functional profiles. And indeed, when we do screening of all the genes that we are able to detect, some ARM genes from ARM family comes between those most abundant ones, as those genes gives resistant to macrolides, lincosomycetes, and streptogramins. We also find genes responsible to resistant to tetracycline and as well beta-lactamase resistant genes. So when we group them by antibiotic classes, the most abundant genes belonged to aminoglycosides classes, followed by tetracycline and lincomycetes resistance. These are antibiotics used heavily in both medicine and agriculture, which points again to connection with the urban and livestock emissions. Altogether, what we see here is that the airborne rhizostome are not only diverse, but also relevant for public health, which makes a strong cause for including air and antimicrobial resistant monitoring frameworks. So in those two studies, we are collecting the air on the high altitude. In the first study here, we're collecting the air as well at the 30 meters of altitude. But what about the ground level air, the one that we breathe every day? So here is a study that have been done for you last year, where the main question that we have is how stable the air microbiome is and whether the neighborhood can influence the composition of air microbiome. In this study, we were the first one actually to implement nanopore sequencing to be able to investigate their composition by this rapid technology. So we selected for us 5 different sites, 2 from transit districts and then 2 from urban background stations, and 1 was kind of rural, but rather rural, I would call it a green environment, where we collected our air samples using the Coriolis technology, and it was extensive air sampling for 2 weeks. We randomized the sampling approach in order to get the representative air microbiome. We were very surprised when we see that each sampling site have its own microbiome. But not only this, but look how stable they are. Each area has its own microbiome, with their particularly the green, so the urban ones, they do characterized by the presence of such a urban organisms, particularly Deinococcus and Paracoccus. Those are characterized for the green spaces, for urban spaces, while the green belt stood out with a unique taxa like Rubrobacter and Myclolatopsis. And those that we detected in the urban sites was not detected in the green belt. When we turn to analysis of the antibiotic resistant genes from those different sites, we found quite a few of them, and surprisingly enough, we had quite a lot of them. And the antibiotic resistant genes presented were distributed across all the sampling sites. And one of the most contaminated, if we could say, environment was the urban beach. And the most important, what we have found in this study is that vancomycin and disinfect resistant genes have been presented mostly in all the sampling sites, demonstrating the extremely importance of being able to track the distribution of those antibiotic-resistant genes within the different districts using the technologies that actually is working and already here. So far I was sharing with you the data we have from the outdoor air, but in parallel, we are working with the indoor air as well within the European project Vienna, where we are aiming to basically characterize the indoor air microbiome and underline that each environment can have its own unique fingerprint. We are aiming to implement the purification of the air in order to improve the air quality. And last but not least, within here, we are aiming to see whether we can build the correlation between the air and human microbiomes. To study the indoor air microbiome, we implemented very similar technology to the one that is used in those previously studies I was discussing. The principle is the same. We are collecting the air with a high flow rate on the filter, and then we work with the filters. This project is quite complex in a way that we are using the entire filter for different type of analysis. So and as well, we're using another MDA8 sampling device for viruses collection. We are using the technologies that very similar one workflow in order to compare the microbiome and human microbiome data. So what we see in the indoor environment, that indeed elderly care center, so this is the data from the elderly care center and the hospital, what we see that there is quite a lot number of those species that are common between those two indoor environments marked in red, but each of those environments, they do have their fingerprints. As such, for elderly care center, we have a dominance of some fungi, particularly those Teletopsis washingtonis and Disconium aciculari. Those fungi has a strong association with the trees. And if we have a look at the surroundings of the elderly care center, we can see that there is quite an intense green environment around. When we talk about the hospital indoor environment, what we see that apart from those pathogens that was already detected, like in the bicycle and very compatible way with the elderly care center, what we see that there are some pathogens that are marked with a red arrow that are not normally detected as airborne, but they have quite a high relevance to the human health, and we are able to detect them in the air. When we check for the resistant profile or resistant genes from the hospital and environment and elderly care center, what we see that the Triaso hospital, so this is Triaso comes from the hospital environment, we see that there is quite a higher number of antibiotic targeted alteration genes that were detected in this environment. As I mentioned previously, so we implement both metagenomic technology to get more advanced metagenomic technology together with more classical or culturomics-based approach. So the same filters that we are collecting from this indoor environment, what we do, we culture them and trying to get those organisms, the culturable one, and in our case, we found like Bacillus and Staphylococcus genera, so the mix of environmental and human associated species, something that we have a very similar tendency with our outdoor samples as in our PNAS study, where we are missing this data in we are missing those organisms in our metagenomic data, but when we culture them, we get them. Among them, we detected clinically relevant species that basically we detected Staphylococcus somnius, different type of Bacillus, particular Sirius, Flaxus, Mycoides, etc. So the profile, what was interesting is that from the same environmental, say from the same room in the elderly care center, we detected two types of Staphylococcus hominis, which was shown in quite different antibiotic resistant profiles. But it was coming from the same air, right? So when we were analyzing this profile, we decided we performed the targeted PCR detection of the genes responsible for those resistant, and it all came negative. This is when we say, let's try to do the whole genome sequencing of those strains, and indeed now there is a work ongoing. Unfortunately, I'm not able to show this data since we are still in the analysis part. We're running the analysis, but what we have seen that some of those organisms were misclassified because this was the mal-identification. And there is a further work ongoing in order to understand the resistance of those strains. I hope I was able to convince you that integrating airborne antimicrobial resistance into existing monitoring framework is not only possible but necessary. The air is far away from being sterile. It's enriched reservoir of microorganisms that carry resistant traits. And these airborne organisms are capable of traveling long distance, crossing geographic and environmental boundaries, and potentially reaching a vulnerable population, as we have seen in the hospital and elderly care center environment. This means that they represent not just environment concern, but actually a real public health risk. By including the air in AMR surveillance, we can detect these threats earlier, understand their movement, and develop better strategy to protect human and environmental health. With that, I'm finishing, and I want to thank all my team who is working on those projects. Without them, nothing would be possible. And thank you very much for all of you for your attention, and I will be happy to take any questions.